Okay, I think we can start now. So, Assalamualaikum warahmatullahi wabarakatuh. Very good morning. So, welcome to our FSG 671 presentation. All right. So, we look forward to listen to your review or to your, if let's say you have been in the lab. So, we would be very happy to, for you to share your finding with us. All right. So, without any delay, so I would like to call the first presenter, so Putri Nur Najwa Noazman. So the title of her presentation will be a review on antioxidant and anti-acetylcholinesterase activities related to Alzheimer's disease in selected Asteraceae species subjected to different extraction parameters. Okay, Putri, so the platform is yours.
Thank you, sir. Can everyone see my slides?
Yes.
Yes.
Thank you. May I start now?
Yes, please.
Alright, Bismillahirrahmanirrahim. Assalamualaikum. Good morning, everyone. My name is Putri Nur Najwa binti Nur Azman. My dearest supervisor is Dr. Wan Rozana Muhammad Hassan. And today it is my pleasure to share with you my humble final year project, which is a review on antioxidant and anti-acetylcholinesterase activities related to Alzheimer's disease in selected Asteraceae species subjected to different extraction parameters. Let us move on to a bit of background study. Alzheimer's disease, or more known as AD, is one of the most common forms of neurodegenerative disease with these clinical symptoms, and it is estimated that the prevalence of AD in Malaysia will increase up to 3% from 2020 to 2050. Next, one of the most important pathologies of AD is the deficit levels of a neurotransmitter called acetylcholine, which is due to the rapid hydrolysis of an enzyme called acetylcholinesterase. Besides that, past literatures have found a strong link between oxidative stress and Alzheimer's disease. When a cell, in this case neurons, are under oxidative stress, it will impede their basic functions. Hence, with the loss of basic functions of neurons coupled with the deficit levels of acetylcholine, it will lead to cognitive decline and other symptoms of Alzheimer's disease. Not to forget that the current frontline drugs that are used for AD treatment have several side effects. One of it would be gastrointestinal discomfort. That is why these days that scientists are looking for safer alternatives for AD treatment in plants. And in this review, I have narrowed my scope of study in 5 selected Asteraceae plants. Why Asteraceae? Because it is reported that they have various medicinal properties like anti-inflammatory, antimicrobial, and other enzyme inhibition. These are some of the research questions that are involved in this review. There are 5 of them here. Moving on to the aim of review. The first aim is to study the relationship between total phenolic and flavonoid content and antioxidant property in selected Asteraceae plants. Number 2 is to analyze effects of various extraction parameters towards anti-ACTH activity in these plants. And lastly, to investigate any possible synergistic dual effects between antioxidant and anti-ACTH. Moving on to the first analysis, which is relationship between total phenolic and flavonoid content with antioxidant property in these selected plants. The results from the 5 plants are summarized here in table 1. Let us move on in depth with the first plant, which is Calendula officinalis, and the first 2 factors to be discussed would be the extraction techniques and the plant parts. In this review, Calendula officinalis is the only one that was extracted from 3 different plant parts using 4 different techniques. Some of the main findings are, number 1, regardless of extraction methods applied, the C. officinalis root methanolic extract yielded the least TPC and TFC value and recorded lowest IC50 value, which makes them stronger antioxidant activity, which did not correlate to most of the findings in this review. Besides that, the leaf methanolic extracts obtained from these 3 extraction methods yielded the highest TFC values among other C. officinalis extracts, which seems to suggest that the leaves of C. officinalis contain high amounts of flavonoids. Moving on to the second plant, which is anhydride fluctuans. In this review, anhydride fluctuans were extracted using 4 different solvents of different polarities. And the main finding is the most polar extract, in this case, the aqueous extract, it yielded the least TPC and TFC among other extracts, and it recorded the highest IC50 value for PPH assay, making them the weaker antioxidant, and vice versa for the less polar extracts. Factor number 4, which is type of extract. In most research, the two types of extract that are extracted are the crude extracts and the essential oil. For the first plant here is Achillea filipendulina. The main finding is the A. filipendulina flower essential oil exhibits stronger antioxidant activity than its leaf essential oil. And the higher the TPC and TFC yielded in crude extract, the stronger the antioxidant activity, which can be observed here in the leaf ethanolic extract. Moving on, still with the same factor, type of extract, but with results from Artemisia herba alba, the main finding is that A. herba alba crude methanol extract exhibited strong antioxidant property than its essential oil. Last but not least, the factor number 5, which is harvesting period. It is just as important as other extraction parameters to be considered in the experiment. The main finding is the antioxidant property varies during the different harvesting period, whereby the extract obtained during the flowering period recorded the highest TPPH activity, which makes them demonstrate the highest antioxidant property among other extracts here. Moving on to analysis number 2, evaluation of ECHE inhibitory activity with regards to different extraction parameters in these plants. The results are summarized here in table 2. Let us move in depth with Calendula officinalis. The first main finding is C. officinalis root methanolic extracts obtained from conventional methods achieved the two highest anti-ECHE activity among other plant parts. Number 2, among all plant parts, flower methanolic extracts obtained recorded all anti-ECHE activity, making it the most versatile plant part. Number 3, the leaf extract that is obtained using ultrasound assisted extraction is the only one that recorded anti-ECHE activity. Moving on to anhydride fluctuans, the less polar extracts recorded the lowest IC50 value, thus possessed stronger anti-ECHE activity and vice versa, as seen here in chloroform extract, which recorded lowest IC50 value. Moving on to factor number 4, which is type of extract. Both plants demonstrated that both crude extract and essential oil have the ability to inhibit the enzyme acetylcholinesterase, but with varying intensity. As we can see here in Achillea filipendulina, the crude ethanolic extract recorded the better anti-ECHE inhibition, whereas for Artemisia herba alba, it is stronger in essential oil. Factor number 5, which is harvesting period, it also plays an important role to influence the anti-ECHE activity, whereby the extract in flowering period obtained recorded the highest inhibitory activity. Last but not least, the possible dual effects. Again, I would like to emphasize that targeting oxidative damage and restoring cholinergic transmission can be an effective approach towards AD, and the results showed that 4 out of 5 plants reviewed showed possible dual effects except for E. herba alba, whereby the crude methanolic extract showed stronger antioxidant property, whereas its essential oil showed stronger anti-ECHE property. In conclusion, all plants showed linear relationship between TPC, TFC, and antioxidant except for Calendula officinalis. Number 2, anti-ECHE activity in selected Asteraceae plants varied significantly, affected by different parameters. And last but not least, 4 out of 5 plants here reviewed showed potential dual effect except for Artemisia herba alba. Regardless, there are 2 limitations that are worth discussing. Number 1 is the lack of information on the standard drug use and its activity. And number 2 is that none of the researchers reviewed here calculated the inhibition factor, which is the inhibitory strength of an extract relative to the reference inhibitor that can be calculated using this formula here. As such, it has led me to a few recommendations. The first recommendation is to address the mentioned limitations in order to come up with a more solid conclusion. Number 2 is to consider investigating different extraction parameters in a single experiment. Since each factor greatly impacts bioactive composition in plants, it is important that we investigate all parameters that can lead to more desirable findings. And last but not least, I would like to recommend for more in vivo studies to be conducted in the near future to better understand how the extracts work on improving acetylcholine levels and cognitive functions. And with that, I thank you.
Thank you, Putri.
Thank you.
So now we are open for Q&A session.
Yes.
Putri,
Yes, Dr. Shirin.
can you go to I think the slide where you showed all of the plants that you are choosing under your study?
Is it this one?
So there are 4 plants in total, if I'm not mistaken, right? 5.
Five, yes.
Okay. So can you just tell me what is the reason why you chose these 5 plants under your scope of study?
Okay, all right. Thank you for the question. So the reason why I chose Asteraceae because it is one of the largest flowering plants that can easily be grown everywhere, anywhere. And besides that, it has been reported that these plants under the species, under the family Asteraceae, have various medicinal properties. For instance, anti-inflammatory, antimicrobial activities, enzyme inhibition, and so much more. Yeah, they have various medicinal properties. That is why I am interested to studying them.
Okay, in your data, did you find anything regarding the antimicrobial activity on
No.
any of these plants?
I have narrowed my scope of study just for the 2 properties, antioxidant and anti-acetylcholinesterase.
Okay, so which one of these is having the highest antioxidant properties based on your study?
Based on my study, I think it's going to be Achillia filipendulina.
All right, okay.
Mm-mm.
All right, thank you.
Thank you.
Putri,
Yes.
those 5 plants that you have selected, is it locally available in Malaysia?
So far as I've read, no, not available in Malaysia.
Time's up.
All right, thank you. So thank you very much to Putri.
Thank you everyone.
All right, good presentation. So now we move on to the second presenter. So Muhammad Haziq Haikal. So Haziq will share with us about his review. So the title will be a review on bacterial PHA extraction method. Okay, Haziq.
Okay. Assalamualaikum warahmatullahi wabarakatuh, and good morning to Mr. Chairperson, honorable timekeeper, and fellow examiners and friends. So my name is Muhammad Haziq Haikal bin Zamal Iswandi, and today I will be presenting on my final year project, my research review on research paper, a review on bacterial PHA extraction method, supervised by Madam Ernie Eileen Rose. So since we are talking about bacterial PHA, so we have to know generally on what is PHA, or polyhydroxyalkanoates, or PHA, is a biopolymer which is produced by many types of bacteria that we can easily find on the sewer, on the soil, such as the E. coli, Bacillus, Megatherium, and Ara eutropha, and so many more. But they synthesize polyhydroxyalkanoate PHA as a carbon store or as an energy source, which is naturally produced when they have lack of certain elements in their surroundings, such as nitrogen or oxygen, and when they have excess supply of carbon dioxide. So yeah, that is why they use PHA as carbon store and energy source when they lack supply. So PHA, this biopolymer produced by this bacteria, can be made into plastics and can be made into conventional plastics we use every day, and can also be used for various biomedical applications, in such as drug delivery. We can use for drugs and tissue engineering scaffolds due to their biocompatibility and biodegradability. So these are the few research questions by doing the review, the aim of this review, what's the gap of the study, the conclusions of this review, and the recommendations after I've done this review. So the objectives of this review is actually to identify few methods of extraction of bacterial PHA, and also to identify the few benefits and the drawbacks of each extraction methods of this bacterial PHA. Now this is the main body for the presentation. We will slightly touch on pre-treatment of cell, which we use to essentially to ease the process of cell disruption, to make it easier for us to extract the bacterial PHA. And then we will be talking about the few of the extraction methods in this research, which is the methods which are used to extract the PHA from the bacterial cells. And then um, uh, there is the analysis of PHA that we have extracted, which is required to evaluate the efficiency of isolation methods of several parameters, such as the purity of the polymer, the recovery of the polymer, and also the molecular weight of the polymer. It has to be tested. All right, so now let's begin with the pre-treatment of bacterial cell. There are actually 4 pre-treatment, but we are going to touch 3 here in this presentation, which is the heat pre-treatment, which is used to denature the genetic material and proteins of the cell. And then it also destabilizes the outer membrane of the bacterial cell, which makes it easier when we are using the extraction method to extract the polymer outside of the bacteria. And then freezing pre-treatment, which uses high energy for this operation. However, but it used to digest the contents of the cell by using sodium disulfide, or for short SDS, and sodium hypochlorite. This freezing treatment can be also used to store the bacterial cell for further use under lower temperature. I'm sorry for the cough. And the third one here is the salt pre-treatment, which makes the cells shrivel and dehydrated by treating the cells with sodium chloride around for 1 hour at 60 degrees Celsius with the 8 kilogram per meter cube density. So that is the pre-treatment of the bacterial cell, which we will use to make it easy to extract the bacteria by using the extraction methods later on. And now the few of the extraction methods that I include in this presentation is the solvent extraction method, which are used to annihilate the endotoxin, in which these are the benefits which it when using the solvent extraction method. Mostly it annihilates the endotoxin in the gram-negative bacteria, while also it uses low energy consumption. However, this extraction method uses high cost, high price, and it is hard to recover the 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 biopolymer, the PHA, because it is hard to remove it from the cell debris. And then the next method here is the digestion method, or divided into many methods actually, but to time bound, we are only going to discuss a few about it. So the first one is chemical digestion method. And the first one we will use sodium hypochlorite to use to the cell, bacterial cell, to digest the non-PHA cellular material of the bacteria. However, this method brings out this method not only just digests the non-PHA cellular materials, it also degrades the polymer itself, the PHA itself. So the recovery for this recovery of the polymer for this method is low, but the advantage is that the recovered polymer is at high purity level. Okay, and then the number 2 is by chemical digestion by using surfactants, which is it breaks the cell membrane of the bacteria, and then it produces the micelles of surfactants that is used, and then phospholipid, which will release the PHA into the solution used. This method, the advantage of this method, however, is it is different from this by using sodium hypochlorite before, where it degrades to the polymer, but the surfactant do not degrade to the polymer. And then, but this method, however, the disadvantages is it makes we recover a low purity of the polymer itself. And then we'll be talking about enzymatic digestion, a high dissolution activities on proteins but low effects on PHA degradation, which tells itself on how on the advantages and disadvantages of this process, enzymatic digestion, which is the advantages is that it makes us it's the recovery rate for the bacterial PHA is high, but the disadvantages is uses high cost since we are using lots of enzyme to degrade the cells. So the discussion and conclusion for the review is generally when we done the solvent extraction method, there are many advancements that has been made by several other researchers on recovering the PHA, such as solvent extraction method, which requires high cost and procedure, where chemicals are also used, which is harmful.
Time's up for presentation.
Okay, thank you. Are you going for Q&A?
Haziq Haikal,
Yes.
can you go to your extraction methods just now? Your extraction method page?
Extraction method, yes.
Yes. All right. My first question is that before I go to this extraction method, my first question is that how important it is to do a pre-treatment before you proceed to the extraction method.
Okay. How important the pretreatment is because is very important because we have to make the cell we have to make the cell very easy to what's the word? It's very easy to destabilize the outer membrane of the cell so that we can easily obtain the pH granules inside the cell itself, so that when we pre-treatment, when the cell undergo the pre-treatment, then the process of this extraction method will be much easier, so that more of the polymer can be extracted from the cell.
All right. Okay. Another question, or more towards like a suggestion. I can
Yes.
see that when you were explaining and making a comparison in the extraction methods, I can see that you have make an analysis on which one have their high recovery, which one have a low recovery, which one is giving you more purity, and things like that. So, but it's not clear because I have to really listen to you one by one. So I would suggest that in your writing, you should have a table, okay, that you have a solvent extraction and digestion method. And then you have list down all of the methods, whether it's chemical or enzymatic digestion. And then you should also put whether it is high or low in terms of purity and in terms of recovery. Yes. Okay. So just one last question. So out of this,
Time's up.
which one is the best
The easiest
method?
the easiest method would be a solvent extraction method.
Solvent extraction method. So in terms of both recovery and purity, is it?
In terms of the easy way to do it, and also yes, the recovery of the PHA polymer.
So there are so many solvent, right? Is there any specific solvent for this method?
There is no specific solvent, but yes.
Time's up.
All right. Okay. Thank you.
All right.
All right, so thank you very much to Shireen and Haziq. Okay,
Thank you.
All right, so we move on to the third presenter. So I would like to call Afza. So Afza will talk about a 2-year retrospective study on clinical-laboratory data for viral meningitis from KTP hospitals in 2016 and 2017. Okay, Afza, you can start now. Yes, please. Boleh as well.
Okay. Oh, sekejap eh. Okay, can you hear me well?
Yes.
Okay. Okay, so may I start? Okay. Assalamualaikum and good morning, greetings to fellow panels and audience. My name is Afzan Christina binti Muhammad Bakari, and today I would like to present my thesis paper with the title of a 2-year retrospective study on clinical-laboratory data for viral meningitis from KPG hospitals in 2016 and 2017. Here is my presentation outline, and I am going to cover all of this. Starting with the introduction of my thesis. So viral meningitis is an inflammation of the fluid and membranes meninges surrounding our brain and spinal cord. The most prevalent pathogens in viral meningitis were enterovirus, followed by herpes simplex virus type 2, type 1, and varicella zoster virus. Patients with viral meningitis usually will manifest with symptoms of viral infection, which then eventually will develop with signs of meningitis such as headache, fever, and stiffness of the skin. As for now, there is limited research that has been done on viral meningitis in Southeast Asia, including Malaysia. How my study is going to be significant for future usage. So we will be able to deduce and identify viral meningitis from other types of meningitis accurately, such as bacterial meningitis, fungal meningitis. Then the next one is to portray the distribution of identified pathogens from detected viral meningitis cases. And the last one is to provide an extended explanation regarding demographic characteristic of patients positive for viral meningitis between 2016 to 2017 in KBG hospitals. So moving on to the objectives of my study. So the first one is to analyze the demographic characteristics of patients positive for viral meningitis. The second one is to determine the diversity of the viral meningitis in KPG hospitals between 2016 and 2017 from the laboratory case report notes. And the last one is to determine a temporal distribution of the detected viral meningitis pathogens monthly throughout 2016 and 2017. Next is literature review of my thesis. So viral meningitis is when the inflammation of the central nervous system infection manifests to the leptomeninges. And the prevalence of viral meningitis according to age or population vaccination status has been estimated to at approximately from 0.26 to 17 cases per 100,000 people, while in the United Kingdom it is estimated about 273 per 100,000 people per year. Then there is no prevalence rate to obtain in Malaysia and neighboring countries, as due to many research are mainly focusing on bacterial meningitis. So the viral pathogens included in this disease are EV, HSV2, HSV1, VZV, B19, AV, EBV, and CMV. The methodology of my thesis. So this retrospective study was approved by the KPJ Clinical Research and Ethics Review Committee and University Technology Mara Research Ethics Committee. A total of 133 laboratory case notes of patients that are suspected of viral meningitis are retrieved from the laboratory information system of KPG Hospital. And the timeline of collections are from 1st January 2016 until 31st December of 2017. do you see my slide, or like is it stuck?
Actually, I'm still seeing literature review
Yeah, okay.
on my screen.
Can I present again? Wait, no, I mean like, wait, 1 second. I just need that. So yeah, this is the methodology that I just explained just now. So moving on to the results of discussion. So to the best of our knowledge, this is the first report that is targeting on circulating viral pathogens that induce viral meningitis in Malaysia from private healthcare facility. So to my notice, there are 3 patients from 133 suspected patients have incomplete data. Thus cannot be elaborated. So from table 4.1, we can see the distribution of patients by age, gender, and sample origin. And most suspected patients are male, and the sex ratio between male and female is 1.28 to 1. And the majority of the suspected patients are from the young adult age group, and most of the sample origin are from the southern region. So this slide is a continuation from the previous one. And table 4.2 is showing the number of patients that are tested positive for viral meningitis. And the total positive cases from 133 suspected patients are 20 people, and the gender with the highest positive case are male. Meanwhile, the regions with highest recorded cases in 2016 is southern region, while in 2017 it is the west region. So according to Medscape, the ratio of infected males to females varies depending on the type of viral infection. And for this result, it is clear that male recorded higher positive cases. And the high case in southern region can be caused due to more PCR testing because they recorded higher suspected patient. But for west region can be contributed due to area with poor sanity, immunity status of residents, and etiological agent. And here is the conclusive result of this thesis, which explain on the circulating viral pathogen. So table 4.3 shows the distribution of viral pathogens in single and dual infections detected by rapid PCR film array differential testing. So EBV infection recorded the highest cases of 8 patients, followed by B19 infection with 3 patients, VZV infection and EV infection with 2 patients each, and for CMV, HSV1, HSV2, AV infection, and also dual infection of CMV and HSV1, they recorded now the number of positive patients recorded are 1 respectively. So the dual infection detected in my result is unique compared to past researchers that are only mainly focusing on single infection of viral meningitis. And the age group with highest number of patients is young adult. And from research by Rice, he also identified that viral meningitis is more common in adults and in children and young adults compared to other age group. And the last one is the temporal distribution. So on table 4.1, it shows temporal distribution of viral meningitis by monthly. And the highest recorded suspected patients are in November 2016. Meanwhile, in 2017, it is in the June and September. But the positive cases are in different months, which are in May and July. And these 2 months is known to be in a hot weather or hot season, where according to Logan et al., during the summer and fall, infection rates of viral meningitis tend to be the highest, which support my finding. And in conclusion, the demographic characteristic of patients positive for viral meningitis was explained in detail. The diversity of the viral meningitis in KPJ Hospital between 2016 and 2017 was able to determine with 8 types of circulating viral pathogen. And temporal distribution of the detected viral meningitis pathogen is high in May and July. And my recommendation, so for better understanding on the trends identified, such as the significant diversification of infection by gender, location, age group and symptoms, a detailed archival data system on demographic, symptoms, and viral pathogens of patients is required to be done by government and private hospitals. And here is my reference for today's presentation, and this is where I am going to end my presentation on this thesis. Open for questions.
Okay, thank you, Afza. So time for Q&A.
Afza,
Yes.
why do you think it's more prevalent in young adults in comparison to the elderly?
Elderly, okay. Thank you for the question. So,
That's the result that you have
Also,
covered, right?
yeah, true. Yeah.
Okay.
Okay. First of all, I would say it is high in young adult because they are more exposed to the viruses because they are active in their life, which is they often go outside. While elderly, they usually stay at. I mean, like not stay at home, but like not frequently going out like as young adult do. So like these viruses are circulating in our air, in our surfaces. So yeah, it depends on that.
All right. Okay. Thank you.
Okay, thank you very much, Afzal.
Thank you.
Right, good one. So now we move on to the next presenter. So I would like to call Murshida Nur Rahma. All right. So Murshida will share with us with the title "The Toxicity Study of Local Instance Premix Coffee Using Mung Beans, right, as a plant bioassay."
Can you hear me?
Yes.
Murshida, we cannot hear you now.
Okay, I'm sorry.
Jangan nervous, relax.
Okay. Assalamualaikum and a very good morning to all the panels and fellow friends. My name is Murshida Nur Rahma binti Abdul Rahman and I'm going to present my final year project, which is on the toxicity study of local instant premix coffee using mung beans, Vigna radiata, as a plant bioassay. So for an introduction, the thriving coffee culture in Malaysia has led to the innovation of new premix coffee products. However, the MOH has released a statement expressing concern due to multiple cases of coffee poisoning after consuming the local premix coffee products, and toxic substances were found in the lab test result. This proved that some companies have deceived the consumers by using misleading advertising to conceal the harmful ingredients. So as you can see here, this is one of the coffee products that has been confiscated by the MOH. Therefore, it is essential for us to carry out a toxicity study to observe the effects of the potentially mutagenic agents that present in the product. And plant bioassay can be used to test this toxicity effect, as it was proven by previous studies to be an excellent bioindicator. This study will be valuable as a guide for further study of the effects caused by the consumption of premix coffee drinks on human health. So the objectives of this study are to evaluate and observe the mutagenic effect of the two selected local premix coffees on the germination and morphologies of mung bean plants when treated with different concentrations of coffee solution at 10% and 20% concentration after different treatment periods. So these are the methodology that I use in the study. And for all the parameters were observed daily for 10 days. So I've selected two different instant local coffee brands, which is a well-known brand, Ali Cafe, and a coffee brand that has been banned by the MOH. It was listed as one of the food products containing scheduled poison. So for results and discussion, firstly, on the plant height of mung beans, we can see there was a significant decrease in mean plant height for the Harimau treatment at both concentrations and the Ali Cafe treatment at 20% concentration as compared to the control group. And the mung bean plants treated with the Harimau treatment at the 20% concentration show the lowest mean plant height. However, it is apparent from the table that there was an increase in plant height recorded for the Ali Cafe treatment at 10% concentration. This better development of mung beans at Ali Cafe treatment at 10% concentration for these parameters and all the parameters that we are going to view later is probably due to the due to the presence of caffeine, as the same effect can be seen by the study from Ansari on the study of the effect of caffeine on mung bean plant, where mung bean grows faster in soil with coffee solutions due to the low amount of caffeine in it. And we can see here that for the 48 hours treatment, the groups treated with Harimau brand could not germinate at all. However, it is slightly different for root length of mung beans, as for the 24 hours treatment, we can see that both the coffee brands show an increase in root length mean as compared to the control group, with the Harimau treatment at both concentrations showing the highest mean root length. And this high mean reading is might probably due to the optimum germinating condition in the Ziploc bag, as the moisture and water content is maintained. And for the 48 hours treatment, we can see a decline as compared to the 24 hours treatment, with the Alikafe treatment at 10% concentration showing the highest mean root length. And still, the groups treated with the Harimau brand still could not germinate in the Ziploc bag even after being left for 3 days to germinate. For the germination and survival percentage of mung beans, we can see that under 24 hours treatment, the Harimau treatments show the lowest germination and survival percentage, while the Alikafe treatment at 10% show the highest germination rate. And still, at the 48 hours treatment, the Alikafe treatment with the same concentration, which is 10%, show the highest germination and survival percentage, while the control group show the lowest germination and survival percentage. For the surface area of the leaf of mung beans, we can see that for the under 24 hours treatment, the Harimau treatments show reduce in mean leaf size as compared to all the other treatments, with the Harimau treatment at a higher concentration, which is 20% concentration, showing the lowest mean reading. And for the 48 hours treatment, we can see that the Alikafe treatments show an increase in the mean leaf size as compared to the control group. For the effect on the physical of cotyledons of mung beans, we can see that for all the mung beans under the 24 hours treatment, almost all the physical of the cotyledons from all the treatments show normal development. However, we can see that for the Alikafe treatment at 20% concentration, the cotyledons show slight red coloration and brown spots. However, we can see that the physical of the cotyledons for under 24 hours treatment, the physical of the cotyledons start to deteriorate, as the morphology of the cotyledons, some of the morphology of the cotyledons for all the treatments show brown spots and wrinkled surface of cotyledons, and red coloration can be seen for the Alikafe treatments at both concentration. While for the physical of the leaf and stem of mung beans, we can see that under 24 hours treatment, the leaf for all the treatments show normal development, only the stem for Alikafe treatments shows slight red coloration compared to all the other treatments. However, for the 48 hours treatment, we can see that the Alikafe treatments show abnormal number of leaves, such as trifoliate leaf, and some plant doesn't even have leaves at all. And for the morphology, we can see that for the Alikafe treatments, the edge of the leaf become wrinkles and brownish for some of the leaf, and we can still see the same coloration as the 24 hours treatment at the Alikafe treatment. So based on all the data recorded, it is apparent that the Harimau treatment has caused significant effect to the mung bean plant development, and it can be observed from the germination of mung bean seeds and the morphologies of the mung bean plants after 10 days. And the effect of the Harimau treatment on plant growth can be seen as early as 24 hours. Even though it was claimed that the ingredients of the Harimau brand were similar to the Alikafe brand, a decline in the mung bean plant development can be observed compared to other treatments. So in conclusion, the mung bean plants treated with the Harimau brand caused significant effect on the plant growth. Even though the Harimau brand's ingredients are similar to the Alikafe brand, the effects of the treatment to mung bean have proven that there might be other unidentified compounds in the instant coffee that might be responsible for the decline in plant growth. It is concluded that these morphological differences might probably due to the result of physiological disturbances caused by the treatment induced to the mung bean seeds. So future research needs to be conducted to study the effect of the premix coffee treatment on the genetic level of the plant and the core effect that has caused the physiological disturbances to the plant. The toxicity test should be carried out in an optimum abiotic condition by adequately monitoring the nutrients supplied to the plants to prevent nutrient deficiency and abiotic stress, as the lack of care for nutrients might also be the cause of the physiological disturbances. So these are all the references that I use for the presentation. Thank you so much for your attention.
Thank you, Murshida. So one question for me, simple one. So a very nice data. Okay.
Thank you.
So from your data, do you think you can increase the public awareness about this issue?
Okay, thank you for the question. In my opinion, my data can increase the awareness of the society for the harm caused by the Harimau brand, as it shows significant effect to the plant growth development as compared to the Alikefe treatment. Thank you.
So do you think, because you have suggested previously you're going to do this genetic testing whatsoever, so do you think it is good for you to know even up to the toxic properties, okay, what type of toxic or toxin can be found in those coffees? So do you think we should do that as well?
Thank you for the question. I think I need to do the cytotoxicity study on this Harimau brand to further know what is the core effect and what effect that it might induce to the chromosome of human.
Right.
Thank you.
So sorry, nak tanya out of curiosity,
Yes.
mungkin saya tak terlepas pandang ke, what exactly caused the kerosakan pada Mung Bean tu? Exactly what elements or ingredients in the harimau tu?
Okay, thank you for your question. The KKM has found tadalafil, which is a scheduled poison in the Harimau brand.
Okay, so it's not the caffeine or whatsoever, right?
Yes.
And may I know why did you choose to compare with Alikefe? I know you said they have the same composition or ingredients. Why you choose Alikefe instead of Nescafe? Because most people drink Nescafe.
I use Alikefe, the exact Alikefe treatment, since the ingredients of the Alikefe brand is almost similar to the ingredient of the Harimau brand. Yes.
Okay, thank you.
Okay, thank you so much for
Okay,
the
Mushida,
question.
one last question, a simple one. If you
Yes.
and your supervisor plan to publish this data, do you think you can expose the name of the product?
Okay, thank you for the question. I'm sorry, but I don't have any answer to that yet. Maybe I will discuss with my supervisor.
All right.
Thank you.
Time's up.
Okay.
Thank you very much, Mushida. So now we move on to the next presenter. So I would like to call Shaza Shakirin. Okay, Shaza will be talking about biosurfactant screening methods of Bacillus subtilis and Pseudomonas aeruginosa.
Okay, thank you. Is the slide visible yet?
Yes.
Yes.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum and a very good morning to lecturers and my friends. I am Shaza Shakirin binti Zainuddin, and today I am going to present my research topic on biosurfactant screening methods of Bacillus subtilis and Pseudomonas aeruginosa. Here are my presentation outline for today's topic. So before going deeper into this topic, I just want to give an idea on what I am going to present today. According to Vijay Sakara, high crude oil demands by various industries have become worrisome. This is because crude oil was listed as one of many sources that is responsible in releasing toxic chemicals, triggering many negative effects towards the environment. Thus, biological approach has caught many eyes to fix this major problem compared to applying chemical or physical approach. Bioremediation is the example of biological approach that uses living organisms such as bacteria, yeast, or fungi in treating contaminants like hydrophobic pollutants. Following that, hydrophobic pollutants are hard to degrade. Therefore, substances known as biosurfactants that are produced by bacteria is a critical characteristic required in playing a beneficial role for bioremediation process. According to Su, Bacillus subtilis and Pseudomonas aeruginosa has shown a good production of biosurfactant and is a suitable candidates for degrading oil. Next, the process of this research was employed into a diagram for a clearer vision. The question that I'm trying to answer here is, how does the biosurfactant production of Pseudomonas aeruginosa compare to Bacillus subtilis? And to answer this, the biosurfactant production manufactured by both bacteria by using the recommended screening methods was observed. The scope has been narrowed down in which the range of interfacial surface reduction will be highlighted. Sorry. Two promising screening methods, which are all-spreading method and tensiometric measurement, were selected for this topic. From the previous articles that I have read, there are several methods to screen the biosurfactant production. However, it has come to a conclusion that all-spreading method and tensiometric measurement were the best, as it is easy to carry out, has a wide range of procedures, and is sensitive. It has also been proven successful from previous studies. Supernatant that was used here for both methods was acquired from the bacteria itself and act as the biosurfactants. For all spreading method, the clear zone formation when the supernatant was poured onto the oil thin layer will be observed. The clear zone formation indicates the presence of biosurfactant. In the other hand, the reduction of surface tension of water will be identified for tensiometric measurement. Moving on to analysis and interpretation, the results for all spreader method that shows the clear zone diameter in centimeter was tabulated. As you can see, for both Bacillus subtilis and Pseudomonas aeruginosa, the minimum clear zone starts at 5.2 centimeter. Chakrabarty stated that microorganisms which has the capability to displace oil up to 4 cm and above were considered a bacteria with good surfactant activity. Therefore, both bacteria shows a positive strong results, preferring to Pseudomonas aeruginosa, which depicted the best surfactant activity, reaching up to 7 cm clear zone diameter. Next is tensiometric measurement. According to Vera, a good examples of biosurfactant producing bacteria are the ones which are able to reduce surface tension below 40 millinewton per meter. While both bacteria, Bacillus subtilis and Pseudomonas aeruginosa, reveal an impressive result, Pseudomonas aeruginosa once again revealed a greater capability compared to Bacillus subtilis that reached the maximum reduction as lowest value at 22.6 millinewton per meter. As for the discussion, both bacteria, which are Bacillus subtilis and Pseudomonas aeruginosa, were selected and studied for their biosurfactant production from diverse points of view. And among these bacteria, Pseudomonas aeruginosa performed well, considering the overall results acquired from the both methods used. Nonetheless, Bacillus subtilis is still an appropriate candidate for bioremediation process. While petroleum has been listed as one of an important sources for industries in Malaysia and therefore were prone to involve with oil pollution, Malaysia still shows a lack of exposure towards the biosurfactant related topic. Thus, local research should indulge more with these topics that will eventually lead to the discovery of other types of bacteria that has the same potential as Pseudomonas aeruginosa and Bacillus subtilis in handling hydrophobic pollution. The study also implies for both academic and practical industries. For academic implications, this study will contribute a theoretical and practical knowledge regarding bioremediation process that demonstrates the influence of biosurfactants on hydrophobic pollutants at laboratory level. The study also exposed the advantages of using biosurfactant as a tool for bioremediation process, in which it is low cost, environmental friendly, and high performance that would be useful for industries in Malaysia as to reach a better future. Last but not least, I have published a poster on junior science communication with the topic an overview of biosurfactant production by Bacillus subtilis and Pseudomonas aeruginosa. In here, I go a bit detail on why biological approach is the top choice compared to applying physicals and chemicals remediation, as physical and chemical radiations excessive usage of harsh chemicals, and because of its production of secondary toxic byproducts apart from being high cost. I also listed some of bacteria that are also capable to produce biosurfactants along with the surfactant classes. And as for biosurfactants, aside from being useful for bioremediation process, it is also it also contributes benefits on food industries, and also can act as antimicrobial activity. Even biosurfactants has got many eyes. It has its own limitation as it relies on several conditions such as temperature, salinity, and pH. Here are my listed reference for this slide. I think that is all from me. Thank you for lending your ears. I hope you guys enjoy.
Okay, thank you very much, Shaza. So just a quick one from me. Okay, so this is a minor comment from your first slide, and then your discussion slide and your poster slide. I think the bacterial species name shouldn't be spelled using caps lock.
Okay.
All right, so that is minor comment. So my question will be quite simple. So at the first place, what makes you to choose those two bacteria? What makes you to choose Bacillus subtilis, okay, not another type of gram-positive, and why do you choose Pseudomonas aeruginosa, not the other type of gram-negative? Is there any special reason for this?
Okay, thank you for the question. As I go through some past articles that I have reviewed, Bacillus subtilis and Pseudomonas aeruginosa was the best studied among other bacteria. And when I go deeper into this both bacteria, Bacillus subtilis produce a surfactant called surfactin, and also Pseudomonas aeruginosa produce a surfactants called rhamnolipids, which contributes a lot on bioremediation process. And it's also, the study about them is also intense, and there are a lot of information that I can get from both bacteria.
All right, thank you. Any more questions? Right, so I think that's it. Okay, thank you, Shaza. Thanks again. Nice one. Okay, so I would like to call the next presenter, so Nur Shakira Atika. So Shakira will talk about antibiotic use and antibiotic resistance in livestock and its environment in Malaysia. So the platform is yours.
Can you hear me, Doctor?
Yes.
Yes, we can hear you.
Can you see my slide?
Not yet.
Not yet. Yeah. Can you see it now?
No. How about the rest? I cannot see it. I just see it's your face.
I cannot see
Yes.
it.
Yeah.
Wait a moment. How about now?
Yes, we can see it. Just put it in presentation mode.
Okay. Assalamualaikum warahmatullahi wabarakatuh, and good morning to everyone. So today I'm going to present my article review entitled Antibiotic Use and Antibiotic Resistance in Livestock and its Environment in Malaysia. My name is Nur Shakira Atika binti Muhammad Rozi, and my supervisor is Dr. Nora Shirin. So what is antibiotic? So antibiotic is defined as a chemical agent that used to kill or inhibit the bacterial growth, while the antibiotic resistant is known as a resistant mechanism acquired by the bacterium against the antibiotic. And livestock or food animals are the animals that are reared and bred to produce food for human. So why are antibiotics are used in the livestock? So the antibiotics are used in the livestock for the treatment of infectious disease in animals, also as a control treatment to treat a group of clinically healthy animals, which to minimize a possible outbreak of the disease, or it also can be used as a prophylaxis, which to prevent those at risk from becoming infected, and as a growth booster to enhance the animal weight. So, however, the improper use of antibiotics to prevent the infection or as a growth promoters has induced the emergence of antibiotic-resistant bacteria in the livestock. Therefore, this is worrying because the antibiotic-resistant bacteria actually can spread from the livestock to human in a various way. For example, through the direct oral route of the meat consumption or through the feces contamination in the food and water. So, this is my research questions of this review. So the first one, what is the scenario of antibiotic resistance within the livestock in Malaysia? And the next one is why awareness of antibiotic use and antibiotic resistance in livestock is essential. The third one, who are the responsible in regulating the antibiotic usage in the livestock? And what are the strategies that can be imposed to control the spread of antibiotic resistant bacteria in the livestock and its environment? And finally, what can be concluded based on the data reviewed from this study? So this is my objective of this review. The objective of this review is to determine the scenario of antibiotic resistance in Malaysian agriculture setting, which focusing on the livestock and its environment based on the previous studies, to highlight the importance of the antibiotic resistance surveillance and to raise awareness regarding antibiotic resistance within the livestock farms, market, and food processing facilities. So the antibiotic use in livestock in Malaysia. So in Malaysia, the usage of antibiotics for the purpose of disease control treatment is being regulated by the National Pharmaceutical Regulatory Agency, NPRA, under the Ministry of Health Malaysia, while the Department of Veterinary Service is the one responsible for the use of the antibiotic in disease prevention and growth promotions under the Ministry of Agriculture. So there are a total of 458 types of antibiotics that are currently being used out of the 688 registered drugs under the NPRA. So some of these antibiotics have been marked by the World Health Organization, WHO, as a critically important antimicrobial. So the critically important antimicrobials that is the antimicrobials that critically important to treat disease in humans. So it should not be used in the livestock. So the prevalence of antibiotic resistance in the livestock, animal excreta, food product, and its environment. So the emergence of antimicrobial resistance is correlated with the usage of antibiotics as the demand and the consumption of poultry are increasing yearly in Malaysia. So there are 7 papers for the prevalence. So 3 out of the 7 papers choose Salmonella species to be isolated. So these are the several species of the Salmonella species that have been reported to be resistant against various types of the antibiotics. So there are some of the critically important antibiotics, which is ampicillin and erythromycin. So Salmonella species is important to be studied because Salmonella species is a pathogen that is commonly associated with the livestock and the foodborne disease. So next species is the Listeria species, where 2 out of the 7 papers choose this bacteria to be isolated. So it's also resistant towards multi antibiotics. So Listeria species is known to cause a zoonotic disease known as a listeriosis. The next is lactic acid bacteria. So there's a various type of lactic acid bacteria that are actually being isolated and also resistant towards a few of antibiotics. So lactic acid bacteria is a little bit different compared to another bacteria in this review. It's because lactic acid bacteria is actually act as a probiotic, and it actually can replace the antibiotic in livestock. However, lactic acid bacteria is still a bacteria, so it have a possibility to be a reservoir for the antibiotic resistant genes, which is significant as the resistance can be transferred through the horizontal gene transfer from one bacteria to another bacteria. The next species is E. coli. So E. coli is important to be studied because it's a commonly found bacteria in both human and livestock. And finally is a Campylobacter species. So Campylobacter species is significant because it causes an infectious foodborne disease known as a Campylobacteriosis. So based on all the data, okay, almost all the it can be seen that almost all of the resistant bacterial species that were reported in the review article were found to be pathogens commonly associated with the foodborne disease. And most of these bacteria were found to be multi-drug resistant. And we can see that most of isolate were observed to be resistant to ampicillins and chloramphenicol. Chloramphenicol is actually one of the restricted antibiotic to be used in the veterinary and the food animals in Malaysia. However, we can still see that chloramphenicol resistant bacteria are detected among the pathogens within the farm, animal excreta, poultry, and meat products. So it can be due to two reasons, which is the first one, it might be due to the misuse of chloramphenicol against the livestock farm practice scheme program in animal farming, or by the transfer of antibiotic resistant genes that could confer a resistance against the chloramphenicol from non-livestock related bacteria to the livestock related bacteria. So it can be concluded that based on this data of this review paper, it shows that the current regulation imposed are not 100% effective to control the situation. So here are some suggestions that recommendation that can be done to improve this. Is the first one to have a regular AR surveillance and more strengthened policy and regulation regarding the non-therapeutic usage of antibiotics in animals farming and agriculture can be imposed. Next is a campaign to raise awareness regarding the antibiotics and antimicrobial resistance should also be increased, targeting not only the farmers, but all levels of the society. So that's all from me. Thank you for listening.
So thank you very much, Shakira.
Thank you, Doctor.
Shakira, one simple question to you. So what will be the limitation and challenges for us to educate the livestock farmers about this issue? Because the same thing happened in anthelmintic resistance, right? So in parasitic drugs, so it is not easy to educate our local farmers. So what will be the challenges with them?
But then, Doctor, I can't hear the question clearly.
Okay.
Can you repeat the question, Doctor?
Right. So the question is very simple. So what can we do to educate? Okay, because this thing will bring challenges for us to educate our local farmers about this antibiotic use and antibiotic resistance. So how can we do that? Because same thing happens in anthelmintic or parasitic drugs resistance. So it is not easy for us to educate farmers. So what can be done in your opinion?
In my opinion, because to be actually the policies, we have a policies nowadays, but I think like what we can do is actually personally approaches the farmers and educate them. I mean like meet them, okay? Because I think what we are doing today is more like we put a regulation, we imply a rules. So the farmers is not feels like for me, I think like it's best that we approach the farmers, so we can educate them. But I say like so what I want to say is actually, for me, I think like the best approach that we actually meet the farmers face by face, or in order to educate them, compared to just like we have the regulation, just follow this regulation.
Correct. That's good. The thing is that you should talk to them and explain to them. Right. So good one. Right. Thank you, Shakira. Is there any more questions before we move on to the next presenter? So
Time's up.
All right. So thank you very much, Shakira. So I'm going to ask you guys, is it okay if you just continue, or you want to take 5 minutes break?
It's okay for me, but you can ask the other students what they think.
Okay, thank you, Shrin. So how about the others? Yati, Judith, students, so do you plan to take 5 minutes break, or you just carry on?
Okay for me, Doctor.
Okay. Yati, macam mana Yati?
Okay, I okay je.
Okay apa? Okay nak break ke? Okay nak
Okay, continue.
Okay, continue.
If the student want to continue, we just continue.
All right, okay. So I think we just continue. So I would like to call the next following presenter, which is Nur Alia Amira. So she will be talking about α-amylase inhibitors, review natural compounds from selected Lamiaceae plants of different extracts related with antidiabetic properties. Okay, Alia.
Thank you, Doctor. Can everyone hear me well?
Yes.
Okay, I will start now. Bismillahirrahmanirrahim. Assalamualaikum and good morning to lecturers and students. My name is Nur Ali Amira. I'm being supervised by Dr. Wan Roziano Muhammad Asal with my topics α-amylase inhibitors, a review of natural compounds from selected Lamiaceae plants of different extracts related with antidiabetic properties. To begin with, I'm going to explain about a disease that is related to my review, which is diabetes. To be simple, diabetes happens when sugar is too high in our body, and it is a group of metabolic disorder from insulin deficiency in secretion, action, or both that are due to hypoglycemia. And it can be controlled by the action of anti-hypoglycemia via inhibition of responsible enzyme in glucose absorption that breaks down the carbohydrate itself, which is alpha-amylase. In addition, the frontline drug, which is acarbose, that used to treat the diabetic patient, unfortunately has unpleasant side effects, which are flatulence and diarrhea. Due to that, natural medication from plant has been pointed out as an alternative to treat the diabetes itself. The plant extracts reported able to inhibit alpha-amylase and reduce the absorption of glucose after the meals. Therefore, 10 plant species in this review from Lamiaceae family were analyzed for their alpha-amylase inhibitory activity and phenolics and flavonoid contents. So, moving on is the 5 research questions that we're going to present, which are what are the aims of this review. Does different solvents and part of the plant's use will affect the alpha-amylase percentage inhibitions? And what are the conclusions and the future recommendations for enzyme inhibitory activity of the plant species studied? Next is the objective of this review. This article review will compare the alpha-amylase inhibitory activity and the phenolics and flavonoid contents of different parts of the plants using different extracts, and also will review the anti-diabetic properties of the plants that give rise to inhibit alpha-amylase activity. Moving on is the plant species of the Lamiaceae family. This is 4 out of 10 species, which are Vitex nangoda L, Zataria multiflora, Ostozia persica, and also Scutella barcellensis.
These 4 species either have pharmacological activities or therapeutic effects. For example, Vitex nangona L has anti-genotoxics, antioxidant, analgesics, and many more. And next is the Scutera bacillensis. It is the species that has flavonoid compounds which exhibit antioxidant properties. Not only that, the next 4 species which are Saturija sculinifolia, Orthosiphon staminospan, Calamita nepeta, L. subnepeta, and also Leucocephalotes. These 4 species has natural medication, has been used as traditional medicine to treat various diseases. For example, is Orthosiphon staminospan. The plants used to treat urinary tract disease, for example, is kidney stones, and Leucocephalotes. It is used for treating many diseases such as asthma, inflammation, paralysis, and even skin disease. The last 2 species that I will going to review is Mentas picata L and Ziziphora taurica subscleronoids. For Ziziphora taurica, it has the studies for this species include antioxidants, antihypertensive, anti-cancer, and even antidiabetic properties itself. Moving on to the α-amylase inhibitory activity and the bioactive compound of the 10 plant species. For the first one, which is Vitex nangona L, it can be seen that it used different parts of the plant, which are leaf and roots, and giving huge difference in alpha-amylase inhibition. It can be seen that root is much lower in inhibit alpha-amylase when compared to the leaf, and it also happens when comparing the total phenolic contents and the total flavonoid contents. Next is the Scutera bacillensis. Which is the IC50 is actually very weak when compared to the standards, and the Zataria multiflora, it used a variety of extracts, and even petroleum ether do not seem to have any reaction inhibit alpha-amylase, and the highest extracts that inhibit alpha-amylase is ethyl acetate, which is 5.5% when compared to other extracts. Moving on to the next 3 species, which is Ostozia persica. Once again, ethyl acetate showing that it inhibit highest alpha-amylase, which is almost 100%, which is the value is 99.4%. And for the species Orthosiphon staminospan, it can be seen that the IC50 is very weak when compared to the Alcabos, but it is the only studies that isolate and identify a single compound, which is sinacentine. Sinacentine is a flavonoid in the plant itself that is believed to give rise inhibit alpha-amylase. The next 3 species is Mentas picata L, Calaminta, and Ziziphora. For Menta and Calaminta, it's only the study only studied the total phenolic contents, and for Ziziphora taurica subscleronoids, water extract is the weakest extract in inhibit alpha-amylase when compared to its alcabos. But and ethyl acetate once again is the highest value, is having the highest IC50 value even compared with the alcabos. As the conclusion, some of the plants in this study showing strong potential of inhibit alpha-amylase, such as ethyl acetate of Ostozia persica. However, some of the plants showing weak inhibitory against alpha-amylase. In addition, the inhibition of alpha-amylase may occur in dose-dependent manner, which is in Leucocephalotes. And even though methanol and ethanol the most used solvent based on review species, ethyl acetate show a good result as a potential solvent based on extraction on several species against alpha-amylase. And different parts of the plant have been used have different value in inhibit alpha-amylase. For example, the Vitex nangona L leaf inhibit alpha-amylase much higher than its roots. For what I can recommend from this study is that standardized solvents are needed to study alpha-amylase inhibitory activity of new potential species that also possess phenolic and flavonoid compound. If a future study about anti-diabetic drugs want to conduct in the future, and further study regarding bioactive compound of the species should be conducted, which means isolation, identification, and characterization of every bioactive compound of the plant may help to formalize the anti-diabetic drugs. This is all the reference that I use for my review article. That's all from me. I end my presentation here. Thank you.
Assalamualaikum, Ardia.
Assalam.
Can you go back to your conclusion? So based on your conclusion, based on your data, so which is again the best solvent
I
that
believe
shows a higher inhibitory activity?
I believe it is ethyl acetate because of the result from the alpha-amylase inhibitions of the Asteraceae persica, which is very high, almost 100%.
So this ethyl acetate is generally a good solvent for all types, or is just only for Asteraceae persica?
There are 3 species that use ethyl acetate that showing having higher alpha-amylase inhibition rather than other solvents.
I see. Okay. All right. Thank you.
Okay.
Okay, thank you very much, Alia. All right, good one. Thank you. So can we move on to the next presenter? So I would like to call Siti Nursaidatul Abdurrahim. So she will talk about methods for identification of Mycosporine-like amino acids.
Okay. Assalamualaikum and good morning to all. Okay, first of all, I would like to apologize as I cannot do live presentation due to my poor internet connection and stability. Therefore, I have prepared a video presentation as suggested by my supervisor and asked for help from my friend to play the video instead. Thank you so much for your understanding.
I cannot hear the sound, the voice of the presentation.
I will play a bit. Nanti bagi tahu ya kalau boleh dengar ke tak.
Still cannot hear it.
Tak boleh dengar sebab dia kena share the sound dengan Google Meet ni kot. Audio dia tu. Kena share sekali dengan audio.
Assalamualaikum and a very good morning to the panel of examiners.
Okey, dengar.
My name is Siti Nursaidatul binti Abdurrahim, and today I will be presenting my final year project, which is an article review with the title Methods for Identification of Mycosporine-like Amino Acids, MAAs. MAAs are colorless and water-soluble compounds that absorb ultraviolet radiation and can be harvested from algae, cyanobacteria, dinoflagellates, diatoms, and also other marine organisms. It serves a function as photoprotection, antioxidant molecule, protection against abiotic stressor, as nitrogen storage, and also it serves as secondary metabolites. MAAs are defined by aminocyclohexanon or aminocyclohexanimidone rings that is conjugated with one or two amino acids. And figure 2 shows some of the basic structure of MAAs that can be found, where the most basic one is carotene with absorption maxima at 320 nanometer, and the addition of functional group hydroxyl and carboxyl will differentiate each MAAs based on their absorption maxima. The research questions for these reviews are: what are the methods being used and each steps for identifying MAAs? Then secondly, what is the importance of the study to the researchers interested in doing research on MAAs? Then what is the new information being used in abstraction method in this study? And what is the most common methods in obtaining MAAs? And lastly, what is the suggestion in improving the method in MAA study? The problem statement is, in order to obtain MAAs, several methods for each steps in identifying MAAs need to be performed, and a method that can guarantee success is needed. And the significance is to help researchers who wants to purify and further explore on the applicability of MAAs to find a suitable method to obtain their desired MAAs. And the objective is to find various methods in cultivating the samples, exposure to ultraviolet radiation, extraction of MAAs, and also detection and characterization of MAAs. And these are overview of the steps, where the first one is we need to cultivate the sample, then expose the sample to ultraviolet radiation, and then we extract the MAAs, and lastly we detect and characterize the MAAs. The first method is cultivation of sample, where we will look at the conditions and media used for the sample. And after analyzing several previous studies, it has been found that for the type of medium, it can be grown on either liquid or agar medium. And all of the previous studies had placed their cultures in the culture room at temperature ranging from 20 to 25 degrees Celsius and illuminate their cultures with cool white fluorescent light. And some studies, for example, Jain et al. And Geda et al. Had supplemented their medium with nutrients and other salts. The next step is to expose the sample to ultraviolet radiation, and in this step, the source of UV radiation and the type of exposure vary between studies. For example, UVB is the most common ultraviolet source, followed by UVA and solar radiation. And in terms of time of exposure, normally previous studies would expose their cultures for 72 hours, and some would take into account the light and dark period. However, exposure with time intervals of 3, 6, 9, and 12 had also been recorded. And for all the previous studies, full white fluorescent light was used as control. Then, in the extraction process, the raw material can either be in powder, pellet, or dried algae crust form. And mostly the cells will be centrifuged before proceeding with any further action. Then the cells will be resuspended in solvent for extraction before undergoing sonication. After that, the lysate will be cleared out and by centrifugation, and the supernatant will be collected and brought to dryness under reduced vacuum or using rotary evaporator. And the dried out residue will be centrifuged before HPLC analysis. The solvents that had been used were methanol, chloroform, and or water with vodka supplements. The last step is to detect and characterize the MAAs. Among the analyses to detect MAAs that had been recorded were HPLC and UHPLC-DAD, QTO-FMS. And the samples were eluted with mobile phase that varies for every study. And based on the previous studies, the detection standard documented were schnorrin, palatin, O5 334, and also mycosporine-like. And the peak identification was done by comparing retention time with these standards. And after MAAs were detected, some studies had further analyzed their MAAs for characterization using LCMS and NMR analysis. This study aims to help researchers who wants to purify and further explore on the applicability of MAAs to find a suitable method for each step by comparing all the methods that have been compiled and their accessibility to the resources in performing those methods. And the study is important so that MAAs researchers will have a compilation of the frequently used and efficient methods in studying MAAs. And the new information that has been gained is that in a recent study, it has been found that drying and consequently dissolving the pellets or samples back will reduce the total MAAs concentrations. In conclusion, for the cultivation step, liquid or agar medium is normally used, but agar medium is more common, and in exposure to ultraviolet step, most studies radiate their cultures with UVB waveband, but for 72 hours. Then in extraction method, the raw material is mostly in powder form. The most frequently used solvent is methanol, and the dried supernatant will be brought to dryness using rotary evaporator. Then, HPLC is usually used in the detection of MAAs. And my suggestion is that researchers need to find ways in order to not have pre-concentration for MAAs so that the total MAA concentration will not be reduced. These are the references for my studies.
Okay, thank you, Saidatul.
Yeah. Thank you.
Okay, can I ask one question?
Yes.
Just now, your slide shows that one of the solvent used, I can't remember by whom. 2021. Okay, the latest. Okay, solvent. Okay, water and volatile supplement.
Yes.
Okay, is there any advantage of this solvent as compared to methanol and chloroform?
After doing some reading on that paper, in the discussion part, it has been found that using water and volatile supplements will take shorter time for the extraction, and also it does not produce any toxic effect as compared to using methanol and other polar solvents.
Okay, thank you, Shaza.
Thank you.
Saidatul, just a quick and simple one. What is the importance of this research or review?
What is the importance of it?
The importance, yes.
Okay, I believe that my study is important to any researchers who wants to do research on MAAs, cultivation or experiments, and they do not have to go through many previous papers just to find that what is the commonly method used, and they will found all the steps and methods in my study.
All right, thank you.
Thank you.
Thank you, Saidatul.
Thank you.
Okay, so now we move on to the next presenter. So we have 4 more to go. So I would like to call Nur Shafika Hadira. So Shafika will talk about neurodegeneration in diabetes, the role of medicinal plants for the treatment.
Can everyone see my screen?
Yes.
Bismillahirrahmanirrahim. Without further ado, I will start my presentation for today. Assalamualaikum, a very good morning to the panels and my fellow friends. Today I would like to present my review article, which is titled Neurodegeneration in Diabetes, the Role of Medicinal Plants for the Treatment. In 2019, 425 million people were diagnosed with diabetes, and the number is predicted to rise to 629 million in 2045. Diabetes is a metabolic disorder that characterized by hyperglycemia. Hyperglycemia is a high blood glucose level. Persistent and chronic hyperglycemia promotes reactive oxygen species, or we call as ROS, generation, and leads to a number of complications involving brain function.
My research cap is the etiology and pathogenesis of diabetes mellitus have extensively been studied. However, the association between diabetes and early alteration in cognitive performance remains unclear. The awareness on the issue is urgently needed as diabetes patient is at high risk of cognitive impairment, including brain disorders such as depression, anxiety, and cognitive dysfunction, which can severely impact the quality of life of patients with diabetes. My research question is, can diabetes damage the brain? Will brain complication in diabetes affect life quality? How natural remedies can ameliorate diabetes-related brain complication? Objective of this study is to discuss the relationship between oxidative stress and diabetes brain complication, and to review the potential of several natural remedies in ameliorating diabetes-related brain complication. Before I go in detail, I will briefly explain the potential mechanism of hyperglycemia-induced brain damage. This hyperglycemia can induce the elevation of reactive oxygen species and reduce the anti-oxidant enzyme, which can further lead to oxidative stress. Oxidative stress can induce the brain cellular damage through various ways such as lipid peroxidation and brain protein oxidation. What is a reactive oxygen species? Reactive oxygen species is a free radical that contains one or more unpaired electrons. This makes them very reactive. In diabetic condition, ROS production mainly from mitochondria. As persistent and chronic hyperglycemia, anti-oxidant enzyme can induce excessive ROS. This is where anti-oxidant enzyme come in handy, as anti-oxidant enzyme is a protective mechanism of brain to protect oxidative damage that causes by ROS. This is three of main anti-oxidant enzyme in the brain: superoxide dismutase, catalase, glutathione peroxidase. Any impaired activity of these antioxidants can compromise neurological function. Oxidative stress in brain. Oxidative stress is a condition resulting from increased production of free radicals or decreased level of antioxidants. The brain is an easy target for excessive oxidative insult because it has abundant lipid content, high energy requirement, and have weak antioxidant capacity. The increased free radical due to hyperglycemia can lead to lipid peroxidation and protein oxidation that can contribute to brain dysfunction. Lipid peroxidation is a mechanism where free radicals attack lipids such as polyunsaturated fatty acid that will form malondialdehyde. Malondialdehyde is a reactive aldehyde that harmful to viability of cell and tissue. This can increase the effect of lipid peroxidation is it can increase the membrane rigidity, decrease activity of membrane-bound enzyme, impairment of membrane receptor, and altered permeability. And if the oxidative damage occurs beyond capacity, it can induce apoptosis and necrosis. Brain is vulnerable to lipid peroxidation as it contains high level of polyunsaturated fatty acid. Lipid is a major component of neuronal membranes such as myelin sheaths that help conduct neuron signaling. Any peroxidation or disruption in lipid can compromise brain function. This is hippocampus. Hippocampus is major role in learning and memory process. Lipid peroxidation can induce apoptosis of hippocampal synapses and neurons. This can result in memory and learning impairment. Next, we will look at protein oxidation. Protein oxidation is a covalent modification of a protein induced by ROS. Oxidation of protein in the presence of glucose is resulting in the formation of advanced glycation and products. The interaction between advanced glycation and products and receptor of AGE resulting in rise of ROS generation. This can lead to modification of numerous gene expression in various types of cells, which can affect protein functions such as receptor, enzyme, transport or structural protein, and neurotransmitter. Few studies have established protein glycation leads to AGEs, are linked to the pathogenesis of neurodegenerative diseases such as Alzheimer disease. Diabetes patient is heavily associated with Alzheimer disease, especially in the elderly. Next, I will review. This is the three herbal plants that I will review. which is gingiva officinale, Centella acetaca, and Ficus litoria. Gingiva officinale, also known as ginger, Centella acetaca, we also known as pagaga. Ficus litoria is mastocytic. This herbal plant contains high antioxidant herbal therapies. First, we will discuss the gingiva officinale brain protective effect, which is a study conducted by Al-Akabawi and Al-Foili, 2014. There's found the depletion of SOD, CAT, and GPS activities in the cerebral cortex hypothalamus of all the experimental group of rats. However, ginger able to increase the antioxidant activity to the normal level. There's also increasing of thiobarbituric acid reactive substances, which is a marker for lipid peroxidation in the cerebral cortex, cerebrum, hippocampus, and hypothalamus of diabetic rats. Fortunately, ginger treatment able to decrease the lipid peroxidation of diabetic rats. Next, we will see at the Centella acetaca brain protective effect, a study conducted by Giri Babu et al. 2014. Centella able to reduce the hippocampal oxidative stress due to the free radical scavenging by help preserve the near normal activity of the antioxidant enzyme, which is SOD, CAT, and GPS. There are also evidence of necrotic changes in the hippocampus of diabetic rats. However, administration of Centella acetaca able to prevent the neuronal degeneration and maintaining the hippocampal function in diabetic rats. Next, we will see Ficus litoria's brain protective effects, a study conducted by Nordiana et al. 2018. We can see there is a remarkable increases of antioxidant enzymes such as SOD and GPS, and decreases of thiobarbituric acid in the brain of rats that treated Ficus litoria. Should I continue or stop?
Continue. Just a few more slides. Please continue.
Huh?
Just continue. You have
Alright.
you have a few more slides.
In the conclusion. Conclusion is excessive RS generation is a potential culprit of a diabetes-related brain complication. Diabetes also causes life-threatening brain damage through various routes, including lipid peroxidation and protein oxidation. Gingiva officinale, Centella acetaca, and Ficus litoria antioxidant activity have the potential to reduce diabetes-related brain complication by improving endogenous antioxidant enzyme. My recommendation is, despite herbal therapies having potential as an alternative to manage diabetes complication, side effect of herbal therapies still remain lacking. Thus, extensive studies on the side effect of herbal therapies are much needed. That's all from me. Thank you so much for listening.
Okay, thank you very much, Shafiqa. Right, a very comprehensive presentation. So I have two comments. So first comment, I really love the ginger image just now. It looks fresh and colorful.
Thank you.
Okay. And second one, because we keep on saying about, talking about treating diabetes. We will always talk about natural products, herbs, and so on. So do you know that currently helminths and bacterial infection can also be used to inhibit type 1 diabetes? So currently people are looking for this, helminth and bacterial infection, which you induce, but it should be a special breed of mice, okay? Not normal mice or rats that you have been using so far. It should be a special breed. So when you induce helminths and bacterial infection, they can also, some products of the helminths and some product of the bacteria, so they can also inhibit type 1 diabetes. But this one is more related to immunological things, right? Okay, so that is only my comment. So any more questions from others?
Time's up.
Okay. Right. So thank you very much. Thanks again.
Thank you so much.
All right. So we move on to Nur Rabiatul Misha. So Rabiatul will talk about potential use of ATR, FAIR as a non-invasive diagnostic tool for the detection of diabetic nephropathy.
Okay, thank you, Doctor. Can everyone see my slides?
Yes.
Yes.
Okay, so is it in the presentation view right now?
Yes.
Okay. Bismillahirrahmanirrahim. Assalamualaikum and very good morning to all the panels and fellow friends. My name is Nurabatul Misha and today I'll be presenting my review research on the potential use of ATR-FTIR as a non-invasive diagnostic tool for the detection of diabetic nephropathy. So here are the outline of my presentation for today. A little bit of the background study. Kidney disease has become a global health issue with the mortality rates ranging between 50% to 80%, and diabetes mellitus is also known to be the major leading cause of kidney problems. In 2020, in Malaysia, the prevalence of having diabetes is recorded at 19.6%. Meanwhile, for diabetic nephropathy, it is recorded much higher at 90.7%. So the problem statement from my article review is that the current clinical diagnosis of diabetic nephropathy requires multiple tests and may involve invasive procedures for sample preparations. Despite all the multiple tests, diabetic nephropathy remains as a global health problem due to the late diagnosis of the diabetic nephropathy. So ATR-FTIR is introduced as a diagnostic tool for rapid diagnosis and measures. Here are the three research questions for my review. First, can ATR-FTIR improve the diagnosis of diabetic nephropathy? Does ATR-FTIR have the potential to be a new, non-invasive, safe and quick tool for diagnosing diabetic nephropathy? And lastly, how ATR-FTIR can reduce the time of diagnosis? This review aims to highlight the potential of attenuated total reflection Fourier transform infrared ATR-FTIR spectroscopy as a non-invasive diagnostic tool for monitoring diabetic nephropathy. Diabetic nephropathy can be subdivided into two conditions, which are acute kidney injury and chronic kidney disease. Acute kidney injury is a severe kidney damage that occurs within a few hours or days caused by accumulation of nitrogenous waste that will lead to an imbalance in fluid and electrolytes. The signs and symptoms for AKI vary in severity from stage 1 to stage 3 that will lead to coma. In stage 1, the small amounts of urine output, swelling of the legs, ankle and around the eyes can be observed. Chronic kidney disease is an accumulation of toxic waste frequently reported in the blood vessels of patients. They can develop problems such as anemia and elevated blood phosphorus levels, as well as complications caused by kidney failures. There are several risk factors that will develop to progression of diabetic nephropathy and can be further divided into two, which are irreversible factors and modifiable factors. Firstly, for the irreversible factors, it consists of age and sex factors, family history, duration of diabetes. In the age and sex factors, kidney function begins to decline at a rate of about 1 milliliter per minute per year after the age of 40. Studies also show that diabetic nephropathy is also more common in men compared to women. This is due to the differences in the hormones in each gender. Next, for the family history, it is related in the genetic information in individuals. The family grouping studies showed that genetic factors are likely to be contributing to a large fraction of diabetic nephropathy prevalence. And lastly, for the duration of diabetes, it is independently associated with the risk of microvascular complication, which greatest risks were observed in the youngest age group with the longest duration of diabetes. Moving on to the modifiable factors, it includes healthy lifestyle, obesity, and smoking. Studies show that regular active physical activities may delay diabetes progression. This is due to the physical activity that can lower the blood pressure and eventually will reduce the kidney impairment. Likewise to the obesity, the healthy dietary patterns can actually prevent onset of diabetes. And lastly, smoking can cause glomerulosclerosis, which is the thickening of glomerular basement membrane that will induce the physiological changes of kidney. Next, we are going to talk about the ATR-FTIR spectroscopy. It is an analytical technique used to identify organic, polymeric, and inorganic materials based on their functional groups. Changes in material composition of biological samples can be observed in the FTIR absorption pattern. During the measurement of a sample, the infrared radiation beam will enter the Michelson interferometer and will be split into two beams and directed into the fixed mirror and moving mirror. Will be combined and then causing interference before directing it to the sample. After directed to the sample, the detector will detect the beam and forming an interferogram. After analyzing in the computer, the spectral information of the chemical composition in the biological samples can be obtained in a spectrum diagram. To explore the potential of using FTIR spectroscopy in monitoring diabetic nephropathy, it requires an understanding of the physiological changes of kidney and the spectral features. So the biological abnormalities of kidney caused by diabetic mellitus is actually at the glomerular capillary wall, where the damage will make it more permeable to larger substances such as protein. The concentration of protein such as albumin in urine is minimal, which is lower than 30 mg due to kidney filtration. But due to the capillary wall damage, high level of protein may present in the urine sample, reaching to above 300 mg per liter of urine produced in 24 hours. This will lead to a condition called macroproteinuria. These are the other important biomarkers that can be obtained from urine samples and their respective functional groups. Urine samples can be collected without the need for invasive techniques, and it can give biologically valuable information such as urea, creatinine, and albumin. So it is an excellent as a diagnostic medium, and FTIR spectroscopy has been used to analyze urine samples. Since biomolecules have chemical bonds, the electric dipole moment can be absorbed through vibration in the mid-infrared region, resulting in detailed biomolecular data in the infrared spectrum. So this is an average FTIR spectrum showing the differences of absorbance spectral bands between the healthy individual and the diabetes patient. This proof that FTIR has enough sensitivity to detect changes in a urine sample from a healthy person to diabetic patients. On top of that, instead of doing multiple tests on various samples, FTIR analysis enables to detect all the related biomarkers for diabetic nephropathy in a single sample in a single test. This FTIR spectrum showing the wavelength for the kidney biomarkers found in urine such as urea, creatinine, and protein. Urea was specifically found in the ranges of 1600 to 1700 wavelength and creatinine in 1400 to 1500 wavelength. Besides, the presence of glucose in urine is the index component for metabolic disorders, especially diabetes. Moving on is the FTIR spectrum of glomerular filtration rate level between a healthy individual and diabetic patient with chronic kidney disease at stage 3 and stage 4. This is a peak analysis where we can see the shifting of the peaks from the healthy person to a diseased person. The peak analysis, which includes the measuring peak shift, is important to quantitate the specific components in a compound. To wrap up my presentation, FTIR spectroscopy is a non-invasive, label-free, and rapid technique that allows for the acquisition of biochemical information about the biomarker's molecular composition with minimal simple preparation. This technique also can provide qualitative, quantitative, and multi-component analysis of the samples. But the existing data only supports the potential use of ATR-FTIR spectroscopy for monitoring kidney function and structure in diabetes. However, there are limitations such as several questions remain unanswered, particularly on how spectral markers help in staging the diabetic nephropathy. Thus, it is conceivable to suggest that the further study on ATR-FTIR should be centered around profiling kidney spectral from patients with different stages of diabetic nephropathy. And the identification of those spectral regions to specific biomarkers will also be beneficial towards developing a new non-invasive and rapid diagnostic tool. So I guess that is all from me. Thank you for listening.
Okay, thank you very much, Rabiatul. Right, so I just only have one quick question. So I'm not really sure whether I have missed your presentation. So what will be the sample that you are going to use for this ATR-FTIR? What will be your sample?
Sorry, can you repeat the question?
What will be your sample?
Oh, so thank you for the question. The sample would be urine, as it is an non-invasive procedure, as you can obtain the urine easily without any invasive procedures.
Right. So do you have to process your sample before introducing to this instrument, or you can just directly use it?
Okay, actually for the reason I use ATR FTIR, I choose ATR FTIR because the concept of ATR FTIR itself, that the sample can directly be put onto the machine to be directly analyzed without any tedious sample preparation.
All right, thank you.
Okay.
Rabiatul, just one quick question out of curiosity. What is the current method that is, like the other alternative method that is currently used other than this ATR FTIR? Like if you are not using ATR FTIR, what are the samples, and also what are the method or machinery that is used to test, to diagnose the N as well?
Okay. Thank you for the question, Doctor. Based on my readings, I've read that other than ATR, FTIR, some research also have been done using saliva samples to see the biomarkers, but using Raman spectroscopy.
I see. All right. Okay. Thank you.
Welcome.
All right. So thank you very much again, Rabiatul.
Okay. Thank you.
Good one. So we have two more students to present. So now we would like to call the second guy for today, so Muhammad Amirul Adli. So he will be presenting about antibacterial properties of jering and kerdas extract. Okay, so I think it is easy for me to call the extract by the common name rather than trying to pronounce this species name, right? So Amirul, the platform is yours.
Thank you very much, sir. Give me a second to present.
No problem. Take your time.
Okay, I would like to begin now. Assalamualaikum and a very good afternoon to all lecturers and my fellow friends. For this presentation, I would like to present my final year project entitled Antibacterial Properties of Archidendron jeringa or jering and Archidendron bubalinum or kerdas extracts under the supervision of Dr. Muarazarina Wan Abdul Razak. The search for natural antibacterial compounds in the past 15 years has been gaining traction due in part to the usage of plant-derived antibacterial compounds that do not show harmful side effects when compared to synthetic existing antibiotics such as chloramphenicol and penicillin. In this study, there were two plant species that were studied. The first one is Archidendron jeringa, or also known as jering. The plant species is native to Southeast Asia, and the local communities has been consuming the leaves as a way to induce urination and also to treat wounds and cuts. The second plant species in this study is Archidendron bubalinum, or also known as kerdas. The kerdas species belongs in the same family as jering, and the kerdas fruits has been consumed as an alternative treatments for diabetes, even though the medicinal properties of kerdas fruits is yet to be proven. And there were a few studies on the antibacterial properties of jering and kerdas. For jering, one study conducted by Abu Bakar et al. Showed that the jering leaves, pods, and seeds extracts managed to exhibit inhibitory effects on tested bacteria in this diffusion test. And for kerdas, there was only one studies that studies the antibacterial effects of kerdas, which is conducted by McKean et al., which shows that the kerdas seed extract did not inhibit the growth of Bacillus cereus, Escherichia coli, and Pseudomonas aeruginosa. And the objectives of this study are to determine the antibacterial activity and also to identify the antibacterial compounds present in the leaves and fruits of Archidendron jeringa and Archidendron bubalinum. And now we move on to the methodology involved in this study. There were 3 types of extracts that were tested. The first one is Kerdas and Jering leaves, and the last one is Jering fruits. And each of the extracts were added with 3 different kinds of solvent, making the grand total number of extracts tested in this study were 9. And each of the extracts were tested against 3 gram-negative and 3 gram-positive bacteria. And as you can see in this flowchart, they highlight yellow boxes indicating 5 tests and analyses that were conducted in this study, and here are the results. So the first test that were conducted was bacterial sensitivity test. There might be a slight delay in the slide. And as you can see in the bacterial sensitivity test, the Jering leaves extract in table 1 showed the best inhibition zone diameter for against all tested bacteria. When compared to the Jering fruit extract in table 2 and Kerdas fruit extract in table 3, the inhibition zone diameter can be seen against certain types of bacteria. As highlighted in the red boxes, the different letters indicating significant differences against tested bacteria only between the same solvents. And the inhibitory effects for each of the extracts were different, and the differences may be attributed to the types of solvents and also the concentration of solvents used for each of the extracts. Now we move on to the results for minimum inhibitory concentration, or MIC, and minimum bactericidal concentration, or MBC determinations. So for the MIC determinations, as you can see after this, the Jering leaves extract in table 4 showed the various MIC values ranged from 250 to 1000 milligram per milliliter. And as you can see in Jering fruit extract in table 5 and Kerdas fruit extract in table 6, the MIC values only exist against certain types of bacteria, ranged from 500 to 1000 milligram per milliliter. Now we move on to the MBC determination, where the Jering leaves extracts in table 7 showed various MBC values against certain types of bacteria, which ranged from 500 to 1000 milligram per milliliter. And as you can see in table 8 and table 9, Jering fruit extracts and Kerdas fruit extracts, respectively, do not exhibit bactericidal effects against tested bacteria. Now, the inhibitory and bactericidal effects of some extracts may not be observed due to the higher concentration of extracts that were needed, which means that more than 1000 milligram per milliliter were required for the extracts to show its antibacterial effects. And this phenomenon can be studied and discussed in a study conducted by Huda et al., which tested various concentrations of cinnamon extracts against Staphylococcus aureus. And the result of the study found that the MBC values of the extract against Staphylococcus aureus is at 40%. Now we move on to the last and final analysis that were conducted in this study, which is phytochemical analysis. And as you may see, the major antibacterial compounds with more than 10% concentration that were detected in this in each of the extracts was benzoic acid 3,4,5 trihydroxymethyl ester in table 10. And as you may also see in table 11 and table 12, there were some major antibacterial compounds that were present in more than 2 types of extract, such as benzene acetic acid. Yet the inhibitory and bactericidal effects for each of the extracts did not exhibit in the same way for each of the extracts due to the different types of solvents and concentration used for each extracts. And in order to improve the antibacterial effects of the extract may be added with antibiotics, as the synergistic effects may assist in improving the antibacterial effects of the extracts. In conclusion, this study is the first study to discuss the MBC values and also the antibacterial compounds that were present in the leaves and fruits of Jering and Kerdas. And among 9 different types of extracts that were tested in this study, The jarring leaves extract in 100% methanol and 50% methanol showed the best inhibitory and bactericidal effects. The jarring leaves may potentially be the new reservoir for the antibacterial compounds as listed in these slides. And for future research, I would recommend future researchers to study the antibacterial effects of jarring and kerdas extracts by using different types of solvents and different types of concentration of the solvents. And I would also recommend future researchers to also combine the extracts with existing antibiotics such as chloramphenicol, penicillin, or erythromycin in order to observe the synergistic effects of the extracts with antibiotics. And here are the references for the article journals that were cited in this presentation. Is it stuck on the same compound in different extract slides?
It's on conclusion recombination. Yeah, it's okay now. It's on reference.
Okay. And with the end of the presentation, I would like to make way for the Q&A session. Thank you very much for listening.
Welcome, Amirul. Good one. So, Amirul, can you go back to your MIC slide? Okay, right. So do you think the value of your MIC is very high?
Could you repeat the question again, sir?
Do you think your MIC value, I would say it is quite high?
For some extracts, the MIC value is quite high. For example, the jarring leaves extract in 100% methanol against Staphylococcus aureus, which is the MIC value is at 1,000 milligram per milliliter. But for some gram-negative bacteria, for example, the values are reasonable.
All right. Can you go to your second last slide, talking about the recommendation? Okay, so you have mentioned about benzoic acid, okay, benzoyl, okay, and so on. So from your reading, do you, have you seen the evidence to say that this compound, okay, has been published before to have these antibacterial properties?
The antibacterial compounds that were listed in the slides has been observed to be detected in certain plants that can inhibit the growth of tested bacteria in their studies.
Okay, so last question. So the plant, is it locally plant or the paper it is not from our Malaysian researcher?
Could you repeat the question again, sir?
I mean, you have mentioned about those plants that can, you can see this benzoic acid whatsoever in that particular plant from that published paper. So my question, is it plants from, is it local plants that easily found in Malaysia or is it outside from Malaysia?
In the study, the plants that were studied were obtained from foreign countries, but the plant itself can be found in Malaysia.
All right.
Time's up.
Okay, thank you very much, Nirul. All right, so now we move on to the last presenter. So I would like to call Nurul Ain Shahira. So Shahira will share with us about cactus plant. Okay, so she will talk about the DNA extraction method for cactus. Okay, Shahira, the platform is yours.
Can you see my slide?
Yes.
Okay.
Yes.
I'll start now. So, Assalamualaikum and a very good morning to all panels and also friends. So my name is Nurul Ashaira binti Muhammad Asri, and today I'll be presenting my final year project with title DNA insertion method for cactus under supervision Dr. Wana Rahayati. So as for my introduction, cactus is actually a type of plant that belongs to family Cactaceae, order Caryophyllus, that more than 2,000 species that come from 130 genera. It is also classified as xerophyte as it has a remarkable adaptation that can cope with extremely challenging adaptation environment. So it is also considered as a type of succulent due to the presence of characteristic of thick and fleshy stem and also leaf tissue that can able to store water. And according to Guerrero et al. 2019, among the succulent members, cactus is considered as highly succulent members due to the most dramatic modification that able to survive in some driest environment by relying on the water that have been stored in the tissue to desiccation. So even all cactus possess similar structure as succulent, but not all succulent considered as cactus due to the presence of areole that can be seen in the red circle in the picture and located at the cactus topple that appear in the circular shape and present as a region area in the body and able to differentiate between cactus and also succulent. So what are the unique features in cactus is the first one is cactus pet or cactus rib, which considered as a modified stem, and it can expand its surface area in order to store a large amount of accumulation of water and also act as main photosynthetic organ. And the next feature will be spine, which is modified leaf, and it used to protect cactus from being eaten by herbivore and also prevent drastic water loss by changing the boundary layers of the cactus. So in my research, I provide 5 research questions. So in this study, I would like to give an insight of DNA extraction protocol from cactus, including the selection of cactus part, sample preparation, method, modification, and also comparison for these 2 types of DNA extraction method, which is CTAC based and also DNA kit. So moving on to next part is among all the cactus part, cactus stem, spine, and also root can be used in DNA extraction method, and cactus stem commonly used in previous study when studying cactus at molecular level due to it can provide good amount of DNA compared to spine and also root. So as we are using different cactus part, so in preparation, sample preparation, it can be slightly differ in its sample preparation in term of its amount of sample use, uses of extraction buffer, and also method of disruption. So to simplify the differences, referring to this table, cactus stem and also cactus root possess almost similar in sample preparation except for the extraction buffer use. Meanwhile, for the cactus spine, possess completely different in terms of its amount of sample use, extraction buffer, and also tissue disruption method. So moving on to next. So when determining genetic issue in molecular study, DNA extraction method that need to be conducted should follow a basic principle, which is the first one is destruction of nuclear membrane and cytoplasm that can be done either physical, chemical, and also enzymatic treatment. And next principle will be purification of DNA from cell lysate, precipitation and DNA purification, rinsing sample using alcohol, and also using low ionic strength solution to dissolve DNA. In previous study, the most common ionic strength solution used was stress EDTA, not only to dissolve DNA but also to prevent DNA from degradation. So in DNA extraction method for cactus, we can use both conventional CTAC-based method and also commercial DNA kit. And for cactus stem, we can use for both method. And meanwhile, for the cactus root, we can only use for conventional CTAC-based method due to it provide low amount of DNA, and using conventional CTAC-based able to reduce the risk of not getting any DNA in DNA extraction method. Whereas for the cactus spine, it is used only in commercial DNA kit, as when preparing sample from cactus spine could be very time consuming. So if using in conventional CTAP base could be in longer process. So to repeat the process, we can use the commercial DNA kit. So this is a protocol for cactus, and some of modification in cactus stem and cactus root could be similar. However, in some modification, it could be slightly differ from each other. So for CTAP base, modification for both cactus stem and cactus root that have been compared with original CTAP is, the first one is a change concentration in sodium chloride, PVP, and also pH of the E buffer. Meanwhile, for modification for that specifically for cactus stem only is the changes concentration of beta mercaptoethanol, usage of proteinase K, and also TE buffer was replaced with HPLC grade water. And the modification that specifically for the cactus root only is changes in the sodium acetate solution. Beta mercaptoethanol was replaced with ethanol, usage of Sarcocell, and also prolonged RNA treatment. So in this study, why require modification or additional on cactus protocol? Because each cactus part used varies enormously in its morphological, and some of special characteristics pertaining to DNA insertion can be considered as highly variable. So when conducting two different DNA insertion method, it could have a major differences in terms of a format and basis of insertion method for these two comparison of DNA insertion method. So for the CTAP base, it is a solution base, and it allows the precipitation of the DNA. Meanwhile, for the DNA insertion kit, it is a silica membrane base, which requires silica to binding with the DNA. So to compare with these two extraction protocol, CTAP base was a most reliable method as it is a solution base, act as cationic detergent, and also able to solubilize lipid cell wall and polysaccharide and also enzyme and protein that prevent DNA from hydrolyze and also preserve DNA from degradation. So when using cactus, different cactus, minor adjustments should be done, which is the first one is a small amount of tissue sample used at range between 0.5 to 8 gram in order to minimize the polysaccharide, secondary metabolite, pectin, mucilage, so that less chemical will be used and less toxicity protocol will be provided. So next adjustment would be usage of high salt CTAP at range 1.4 mol to 5 mol. That will be adjusted according to sample use in order to increase the DNA extraction process. And the last adjustment is increased centrifugation and incubation process. So to conclude, both commercial DNA kit and conventional CTAP base show successful in isolating DNA through colorless palette that usually fulfill the criteria without additional process. To compare these two methods, it provides better DNA quality and quantity as compared to commercial DNA kit. However, commercial DNA kit still able to isolate DNA and still falls at acceptable range. So my suggestion in future study, only use small amount of sample in DNA insertion process as it capable to obtain DNA along with fewer chemical will be used and lead to the cost effective protocol. Till then, I end my presentation. So thank you.
Thank you very much to Shaira. All right.
Nur Ain,
Yes.
can you show me one of your slides where you have the modification regarding the DNA extraction just now?
Okay. Is it this one?
Yes. Okay.
Okay.
So in this CTAP-based protocol, you mentioned that there's an additional modification where you where one of the study suggested to add beta mercaptoethanol. Do you know what is the purpose of beta mercaptoethanol?
Beta mercaptoethanol is actually to remove pectin in during the DNA insertion process, so that minimize the contamination during the DNA extraction process and able to obtain DNA.
All right. Okay. Thank you.
Any more questions?
So based on your result, the CTEP is much more efficient to get for a high yield extraction, but with to deal with a smaller sample, the commercial is still okay. Is it? Is that what you have concluded based on your study?
Yes, because for commercial DNA, if it is it able to obtain high yield if when using small amount of samples. So when we conducted in large sample, it could be a limitation to commercial DNA kit.
Time's up.
All right. Okay. Thank you.
Okay, thank you.
All right, so thank you very much to all the students. So we have come to an end of the session. So personally, I would like to congratulate each one of you for giving us a very good presentation despite of this pandemic learning situation. So you all has provided a good one to us, a comprehensive presentation. So there are so many new things that me myself, new things for me to learn for today. So is there any comments from the examiners, all three of you, regarding today's session?
Thank you to the students for a very good presentation. Like what Sir Shamsul just mentioned just now, everyone have done their best. So I think that's it from me.
Thank you.
Yes, I too want to congratulate the students, and I'm impressed with the level of English from this program. Very good, and I wish all the best. Thank you.
Thank you.
Thank you for all students for giving their best.
All right. So I think before we end the session, so I think people would like to see each one of us. So I would like to call everybody to switch on the camera if you don't have a problem with your camera. So at least we can have a photo to be taken, and then I can share with our group later. Masa semua tak senyum ni, kena senyum lah. Dah habis present lah. Serius lah semua. Sorry lah, I punya kamera selalu akan ada masalah. So, might apologize.
Shamsul, you kena kira baru kita senyum. Kita tak boleh senyum nanti penat lah.
Boleh ke?
Kena kira baru dia orang prepare untuk senyum.
Sorry, tak on camera. My profile pic is better.
It's okay, Dr. Judith. Your profile picture is nice enough.
Itulah.
Semua orang takut, dia kata garang. That's why I remain lah gambar. Alright, so ready ya? So smile everyone. 1, 2, 3. Okay, one more. 1, 2, 3. Alright, okay. So with that, again, thank you very much to everyone. Thank you to Shireen, thank you to Judith, thank you to Yati. All right, so thank you to all the students. So you have done a great job. All right, so stay safe, take care. So nothing more to say. So you dah present dan tinggal untuk kami nak bagi markah you punya written thesis or written review. And then that's all, you can have a good proper peace of mind, I would say, especially for this FSG 671. Okay, so stay safe everyone. Thank you very much. Okay, so thanks again to Shireen, Judith and Yati. All right, thanks for your time.
Thank you, Shamsul, Shireen.
All
right, thank you.
Thank you, students.
Thank you so much, everyone. Take care. Assalamualaikum.
Thank you, doctors.
Thank you, doctor.
Bye.
Thank you, doctor.
Thank you, doctor.
Bye. Okay, all right.